So to kick it off, I'm going to start with some background on our company, Visicall, where we came from, what we learned through the I-Corps program, and what we're doing about it today. So for some background on our company, we founded the company about 4 years ago, and we initially founded the company at the exact same time that we entered into the original NSF grant that we had, which was associated with an I-Corps. And our original question was around reagents and software that we had developed for imaging tissues. Essentially, our company is focused on transforming tissues, whether they're mouse brains or biopsies, into large data sets and then mining those data sets for some kind of actionable insight. And through our original I-Corps grant, we learned about what types of customers we should target, what types of technologies and research questions they had. And over the last 3.5 years, we've built a very profitable company. We have a team of 15 employees now. We're leveraging our original technology that we developed through our NSF grant and using a lot of the lessons from our original I-Corps program with all of our customers that we currently work with. So we work with 12 of the top 20 pharma companies, helping them transform their tissues into large data sets and finding answers to different research questions from those data sets. So we have this core technology around how to image tissues and how to image tissues in 3 dimensions instead of traditional slide-based histology. And we operate today primarily as a contract research organization, primarily in the preclinical space. But the goal of our company has always been to take our tissue clearing and tissue imaging and tissue labeling and tissue analysis approaches and apply those to some kind of clinical question. We're helping a lot of pharma companies today accelerate their drug discovery and development efforts, but we really wanted to go ahead and figure out if our technologies could actually improve the lives of patients directly as a diagnostic tool. And that's why we came back to I-Corps. We came back to the NIH program, specifically looking at the clinical applications of our technology platform. Now, I mentioned this a little bit. So where we started was developing technologies for imaging tissues, turning them into 3D data sets, and then also artificial intelligence-based image analysis for taking those large data sets and distilling them down to something useful. Our pharma clients do not care about the technology. They don't care about how the sausage is made. What they're really interested in is those insights that we can pull from tissues. And you'll hear this again and again during the I-Corps process, but the technology doesn't really matter to a lot of your customers. It's really what their problem is and your ability to answer that problem. A lot of times they do not care. Actually, most of the time, they don't really care about the technology, the nuts and bolts of it that much. They care about the answer and the solution to their problem. And that's why a lot of the interviews are focused on your customers' problems and not your technology. So our problem coming into the I-Corps program this time around was that we had this great technology for turning tissues into data and mining that data, but we didn't really know the specific use case for where in the clinic that's actually important. We kind of theorized that, hey, it's probably important in breast cancer. It's a disease that affects a lot of people. We think that 3D imaging might improve the diagnosis paradigm, might get rid of some of our false positives and false negatives, but we didn't really know. We had a solution really looking for a problem. So we had no idea what clinical value our technology would have for patients. We did not understand if our test was possible with the current pathology workflow if we developed a diagnostic tool. We had no understanding of how much a test like ours could even cost. So price and cost are really important to understand when bringing something to market in the clinic, and also how payers actually pay for something and what they're willing to pay. And then lastly, we did not have a reliable source of tissues and data for the clinic, which I'll come back to in a moment. But our main I-Corps goal was to identify how our tool could improve patient outcomes and if it could do so in a financially viable manner. So like Edmund mentioned, the technical piece actually is a very small piece. The big piece is, can you turn this into a business? Can you solve a problem, and will enough people pay for that solution to your problem? So those are our really core questions here with I-Corps. So early on in the process, we got out of the building and started interviewing pathologists and clinical oncologists and trying to understand where our test could add value and if it could actually add value in the breast cancer field. And here's just one interview response that I've highlighted from Ciaran Mannion from Hackensack University Medical Center. And he explained really for us that we should work backwards. We shouldn't say, hey, we're going to image breast cancer tissues in 3D and improve diagnosis. We should work backwards and say, here's a problem. These patients are getting chemotherapy today and they shouldn't. Or these patients should be getting Keytruda or some other second-line treatment and they're not. And then work backwards from there and come up with an application where we're actually changing the clinical practice. If you're more accurate in diagnosis but you're not changing what happens to the patient, it provides no value whatsoever. And that was a learning that we really didn't understand. It seems very obvious now, but you do need to change what's happening in the clinic, and you do need to improve the patient's outcome, or else no one's going to pay for your diagnostic test. So this is where our focus changed. We were originally looking at primary tumors, and then we started looking at sentinel lymph nodes. Sentinel lymph nodes are used to understand if cancer is spreading throughout the body, and if so, the treatment paradigm changes for that patient. So a lot of times for different types of cancers where metastasis is an issue, like breast cancer or melanoma or colorectal, you're pulling out sentinel lymph nodes, which are adjacent lymph nodes to the primary tumor site, and you're trying to see if there's cancer cells in those sentinel lymph nodes. But if you're slicing those into a few ultra-thin slices, you might miss some of those cancer cells. So we hypothesized that if we can look at these sentinel lymph nodes, we could better determine which patients had metastatic breast cancer and improve the outcome for those patients. But after interviewing some folks specifically in this space, we found out that in the context of breast cancer, reducing false negatives for sentinel lymph nodes doesn't actually change the patient outcome unless the metastases are huge. So they're, in the space of breast cancer, if you have a few cancer cells in a sentinel lymph node, it doesn't really change what happens to the patient. So therefore, our three-dimensional imaging and analysis approach could detect a very small number of cells, which is cool, but it doesn't actually change what happens to the patient. And because of that, we realized that our focus couldn't actually be breast cancer, and it couldn't be sentinel lymph nodes. We would provide absolutely no value in this space. But in interviewing folks at Cedars-Sinai, we also spoke with a few oncologists and pathologists, and what they pointed out to us was that while in the context of breast cancer and sentinel lymph nodes, that those few cells don't really have an impact on patient treatment, in the context of melanoma, they actually dramatically change what happens to the patient. So if you have a few cells in that sentinel lymph node, it does actually change what happens to the patient as far as their treatment practice. And false negatives are a large problem in the melanoma space. So through this interview process, we determined first that breast cancer would not be the key space to go into, but furthermore, we identified an area where the same approach, the same technology would provide the clinical value that we were looking for, which was crucial to our I-Core journey here. So just to summarize what this looks like, today, depending upon what data you look at, between 5 to 20% of the time, our sentinel lymph nodes are producing false negatives in melanoma. So you're telling a patient you don't have metastatic melanoma when in fact they do. It's a very aggressive disease, and we want to treat that, and we want to have that treatment paradigm associated with how aggressive that patient's cancer is. With our workflow, what we would do is we would take tissues after they're visualized today. They would be sent to our lab, and we would visualize those in 3D and actually look at each and every cell within one of these large sentinel lymph nodes using our 3D imaging approach. What happens is if we're able to identify patients that are told that they're negative today, that they're actually positive, those patients will then get chemotherapy and more aggressive second-line treatments instead of just passive monitoring. And over a 5-year or 10-year span, this increases life survival by half a year on a 5-year basis and 3 years on a 10-year basis, which is a very, very significant improvement in quality of life and also survival for these patients. So by these interviews, we found a very significant problem that could be addressed with our technology by just interviewing a lot of folks in this space and asking questions about what problems are out there today. This slide is a bit busy, but one of the core questions we had, and as a PhD scientist with no background in the clinical space or payers and payees, figuring out how much we could actually charge for this test was something that was incredibly abstract to us at the beginning of this process. But what we learned is if there's 3,000 or 4,000 patients today that are being treated, that have false negatives and they're not being treated appropriately, those patients each on a 10-year basis are losing around 3 years of life. And the way that we value life in the U.S. for a lot of reimbursement is actually valuing a year of quality adjusted life years at about $100,000 per year. So if we're able to pick up all those false negatives, we can actually save 9,700 years of life, which is about $1 billion in value that we're adding back to the healthcare economy here. And what we realize is that that number allows us to figure out exactly how much we can charge for our test and also exactly how accurate our test needs to be. So as we're thinking about a phase 2 project, we know what our endpoints are based upon what we can charge for this test. So if we're only 10% accurate in picking up those 3,000 false negatives, we probably don't have a financially viable test. But if we can pick up 50 or 60 or 70% of those, we start to have a test that's financially viable and that insurance companies will actually pay for. And furthermore, we can justify the cost of this test to insurance companies based upon the value that's being added to the healthcare ecosystem. So this was a key finding for us. This allowed us to create not only the business model, but actually the specific aims for our phase 2 grant that we just submitted, because we know if we don't hit certain benchmarks, our test can't actually be financially viable. So we're able to pull the science and actually turn it into a reimbursement amount for our phase 2 project. So just to summarize this a little bit, if every patient will pay out of pocket $1,000 per test, we determined through a lot of our interviews that pretty much most patients will actually pay for the test out of pocket. But we determined based upon the value that we're bringing to the healthcare economy here that if we're able to go ahead and move a test through insurance, insurance companies will reimburse between $2,000 and $4,000 for a test like this. So the actual market was also able to be generated through this process, and we determined that if we're to go through reimbursement with insurance companies, the market for our test is around $64 to $128 million per year. But if we're able to go, if we want to go through a cash approach, it's about $32 million per year. So this also allowed us to shape our thinking around our commercial plan. So do we want to go with reimbursement? Do we want to go with cash, so patients paying out of pocket, and get to market much faster, but with a smaller market? So a lot of this thinking came out of our interviews, being able to figure out exactly the market, exactly how much we can be reimbursed, and exactly what the approach is that we're going to go to market with. And then furthermore, like I mentioned, this really drove our endpoints for our phase 2 study. And we can show on this graph here, this table, the accuracy of a test from 0 to 100% and the false negative rate. So if the false negative rate is 1% or 15%, we can figure out exactly how much we can charge for the test. And furthermore, if we're only able to get a 30% accuracy, we start to realize that this test is not going to be financially viable. So we're able to actually draw very quantitative numbers into our phase 2 study about what the endpoints need to be to both create a clinically and also financially viable test for patients, and a test that payers will actually pay for and reimburse for. And then lastly here, through the project, one of the key things that we found by just going out and speaking with a lot of folks was a problem was the data generation and the tissue generation. So having tissues, sentinel lymph node biopsy tissues, is crucial because we'll need to do a retrospective study to prove that this approach actually works and to demonstrate what the accuracy of the test is and how many cancer cells we can pick up. But with that tissue, we also need the associated data. And having that data paired with the tissue was something that was very challenging to find, but through this I-Corps process, we were able to identify Cedars-Sinai, Baylor, and UConn that had these tissues that we needed with the associated data that we needed. And for our phase 2 project, we actually put together a partnership, a subawardee with Cedars-Sinai, who we met through the I-Corps program, and we started to write our phase 2 grant with, and ultimately submitted a few weeks ago now with Cedars-Sinai to partner using their tissues and their data for this project. So not only did we get a lot of insights from Cedars-Sinai, we also got a partner for which we could submit our phase 2 project with and move this project forward, which is crucial to going ahead with a retrospective study. Our next steps coming out of I-Core, the immediate next steps were to go ahead and quantitatively determine the number of images and tissue volume to image to identify a false negative sentinel lymph node biopsy, which we actually worked on after the I-Core process. So our original phase 1 grant was focused on breast cancer. So we went back to the drawing board and repeated a lot of our phase 1 specific aims with melanoma tissues, and then did some additional R&D to get ready for our phase 2 submission. We also purchased a new confocal microscope for our lab. We filed some provisional patent applications on tissue processing, image analysis, and also some labeling approaches. And then we funded all these efforts through our current business profits. So our current underlying CRO business is profitable. So we're able to fund these efforts through that business to get ready for our phase 2 and diving into this clinical retrospective study, which is what our phase 2 is focused on, is demonstrating through a retrospective study with 170 negative sentinel lymph nodes and also some positive controls that we can identify false negatives in a reliable manner, and then also build the software and the infrastructure to do this as a service, and eventually launch a CLIA test to go ahead and characterize these melanoma sentinel lymph node biopsies and improve the diagnosis of disease. So the I-Core process for us was really crucial in putting all these pieces together. We changed our disease focus, we changed our approach, we found a partner, and really got into the details of how much the test could cost, what the market was, and exactly how we should go to market with patients paying out of pocket or patients being reimbursed and going for a CPT code. So this whole process really, really shaped our thinking, and we were able to throw out a lot of the preconceived notions that we had about where we'd fit into the marketplace. So we learned a lot. We've done the process twice, and each time it's really shaped our thinking and pushed us through these interviews to really change the way we think about our business. So with that, I'll leave it for later if there's any questions, or you can contact us at VisiCall for any questions that you have about the process or generally imaging or melanoma in more detail. So thank you.